Buy some drinks, a cheese plate. Please help me welcome Dr. C. McKeon. C has a very interesting background. He completed his undergrad at the Evergreen State College in Washington State, did his master's on poison dart frogs in the Peruvian Amazon, and completed his PhD through the University of Florida on his work in the Central Pacific Islands. He now works as a scientist for the Smithsonian Institution on the ecology of biodiverse marine communities. Please give a warm Sea&Learn welcome to C. McKeon.
You said that out loud.
Didn't think that. All right. So I'll be talking to you today about biodiversity in ecological interactions and how those two things go together. Specifically looking at reef crustaceans. So this is a group, crustaceans is a huge group. Crustaceans now include insects if you think about pan-crustaceans. So that means that a huge percentage of the macrofauna, so things above a couple millimeters in size, actually are crustaceans. We're surrounded by crustaceans. They rule the planet quite literally. But the ones I'm going to be talking to are rather innocent, so let's see. But first we need to start with a few definitions. What is biodiversity? And biodiversity is basically everything. It ranges from environmental diversity, landscape diversity, down to organismal diversity, which is what most of us think of when we're thinking about biodiversity. But also genetic diversity, so within a species. Biodiversity is part of that, the genetic variation in everything, us included. How about behavioral diversity? So the way things act, this also can be incorporated into biodiversity. Huge topic. And what do we get from biodiversity? Pretty much everything. Food, medicine, natural cycles, so the carbon cycle relies on biodiversity, nitrogen cycle relies on biodiversity, ecosystem services, so biological resources, social benefits, all of it is derived from biodiversity, with 40% of the world's economy coming from biological resources. 40%. And then this last factor of resilience. So diverse communities recover faster. I'll be talking a little bit about that today. Everyone understands difference, right? So this is it's an inherent human characteristic, is when we look at different things, we perceive difference very easily. And it's the root of why we collect coins or stamps, is because we have this inherent value on a diverse array of whatever it is that interests you. So this inherent valuation of diversity has been termed by E.O. Wilson as biophilia. So this love for biology. But there's a lingering question of, do we need all these species? Do they all provide some essential service in this biodiverse economy, if you will? And when you think that we have 1 to 20 million, somewhere in there, we don't know the answer to how many species there actually are. We think it's between 1 and 20 million just macrofauna species, things above a couple millimeters. That's an awful lot of species to have an individual effect on that economy, or at least for us to perceive an individual effect. So the other part of this diversity ecology talk is ecology. And what is ecology? Ecology is the interactions between an organism and its environment. And you have to think that every species is interacting with its environment somehow. Either it is being eaten, it is eating, it is living on a rock, it is living under a coral. So all of these are ecological interactions. And every species is going to have its own story. So combining these two major themes of biodiversity and ecology is kind of a tough thing to do. So a lot of what we do is actually modeling. And sadly, scientific models are not what you might think. They are not ridiculously beautiful people who do science. For the rest of us scientists, it's a little sad. More often, scientific and ecological models are up here in this corner. They are math. They're mathematic machines. But they're useful tools. And so when I'm thinking about models and when I'm speaking about models, I don't want you thinking about the math. I want you thinking about toy planes, right? Because you learn something from this. That's a model. You can learn about flight on a very simple, from a plane, right? You slide the wings in, you throw it, you're learning about flight. That is a model of flight. You can make your model more complex. Maybe it'll teach you more about flight, but never are we getting to reality with these models. So this is why you're going to often hear scientists when they're on the news talking about climate change, they say, well, I don't really know, but the model predicts this. So models are tools that we use. They're machines that can help us generate hypotheses in science. Everybody with me so far? Yes. Ditch math. So I'm giving you a model of biodiversity today, all right? And that model is one that I think most of you already know. How many of you have played the card game Uno? Please raise your hand. All right, and if not Uno, then Crazy Eights or something similar. These are games that are, we need to just cross out the name Uno and say biological model of biodiversity. Because this is a great model. And within this model, we can test two different hypotheses, two different questions. The first, well, let's review Uno first. So you're presented in Uno with an ecological or environmental circumstance. You're dealt a green 2. So you have to respond to that green 2 in the game, either with a green or with a 2, right? So if we were to have all greens, you would have what's called functional redundancy. So the 2 is acting in generally the same way as the 1 and the 3 and the 4 in the case of greens. The contrasting hypothesis is one of functional diversity, where each one of our cards, 1 through 4, is providing a different color role. All right? So tonight I'm going to be talking about both of these and talking about testing these two hypotheses on reef crustaceans. So functional redundancy, basically can we take one species, if we get rid of it, then another will slide in to take its place. Or functional diversity, where each one of those species is of pressing conservation importance. All right? Do we have to preserve all species to have a functioning ecosystem? That's what I'm trying to get at. So starting question, which hand would you choose? You're playing this game, you can choose functional redundancy hand or you can choose functional diversity hand. All right, we're going to take a vote. All in favor of functional redundancy? 1, 2. And how about functional diversity? Okay, so we've got some people voting for functional diversity. When you get home, you have to try this. Try both in an actual game of Uno. You're dealt wild cards in life, right? So our responses have to deal with the immediate situation in our environment. So wild cards on a reef are sedimentation or predation, all these different things, algal overgrowth. So you're dealt with a situation, you have to respond to it. And so that's kind of what I'm testing. And the best way to test these things are looking at species that don't move around too much, that you can find really easily. And it just happens that these beautiful coral reefs that I really enjoy spending time on, almost all structural species on a reef have symbionts living with them. Not just the endosymbionts that you're going to hear from later on in the week, but also exosymbionts. So that's mostly what I study. Like here, if you go out and you look at our nice giant anemones, frequently they have different types of anemone shrimp with them. They're obligate associates of anemones. They hang out with them all the time. You can find them. If you find the anemone, you find the shrimp. In the South Pacific, you find a nice big gorgonian, and they've got this wonderful little crab on them called Xenocarcinus, which is a great name. You just have to say it as frequently as you can. Xenocarcinus. And the best tests of functional diversity are closely related species of similar body size and shape. And it just happens that coral symbiotic crustaceans often fulfill this requirement. They also happen to be beautiful and live on coral reefs, both also drivers to me working on them. So the first crab I'm going to be talking to you about is called Trapezia. And Trapezia is found across the Indo-West Pacific. It's an obligate associate of polyploid corals. So these are cauliflower corals, sometimes they're called. We've got currently 23 described species. They're all very similar in morphology. They range in size from about the size of your smallest fingernail to 2 thumbnails across the carapace. So not very big. They occur in pairs. They have this great habit of living as male-female pairs, and these pairs will defend their home against other members of their same species. But they will allow for other species of Trapezia to join them in the coral. So you can have up to 5 species of Trapezia, of the same genus of crab, in a single coral colony. So this is important. They occur in the same place, same time. They're roughly the same shape, same size, roughly. But do they act differently? Do they provide different services? So this was the type of question that I was really interested in asking. So as a first stressor, as a first environmental situation, sediment. Sediment frequently is a stressor to reefs. We see sediment go on to living corals, and it will either cause bleaching or outright mortality. And the crabs will go around like little good housekeepers, and they'll remove all the sediment grains. So they're actually cleaning their corals. But do they do it the same? For this I went to the island of Ofu in American Samoa. And Ofu, for those of you who don't know it, is a spectacular lagoon. It is a hard bench reef. So that means that the waves have scoured out these pools behind the reef itself. So it's a snorkeler's paradise. It's too shallow to dive, but it's wonderful to conduct ecological research in because you can just reach in and move things around. It's hyper diverse, it's exquisite, and cheap. And so my question was, is there functional diversity among Trapezia in terms of their sediment removal? So how do you go about testing this? All right. So what I did, and with permits, everything here was fully permitted, I would go out and I'd remove small corals, about the size of your fist, with their pairs of Trapezia in them. And I would put them into a tray on this little bridge, this little PVC bridge, and I would have the crabs in there, and I would add sediment. The crabs would remove the sediment, literally just throw it off, and it would fall into the bottom of the tray. So I could come back the next morning, pull out the tray, weigh the sediment again, and know how much the crabs were actually removing. So, brief primer and statistics here. All you need to know, I'll talk about this, but all you need to know is if the dots on 2 different columns are the same color, it means there's no difference. If they're different colors, there's a statistically significant difference. All right? Everybody with me? Nod and smile. Okay. So, when I looked at 3 different species of Trapezia, all the same size, about your thumbnail, and then control, so the coral alone, we detected differences between these 2 species of Trapezia. This one's a bit of a slacker in comparison to his cousins. But this one, sometimes it really slacks off, sometimes it's really quite active. So, I've put down that we had differences and that I've said this is a vote for functional diversity. You could read this either way. But it's also important to note that this is in comparison to the control coral. And this is with a large size sediment. So sediment grains that the crabs could actually come up and grab and throw. When I did the same experiment with the two most effective species, what I saw was that there was no difference. All blues across the top here. So the coral by itself could remove just as much sediment as the crabs did. So this is smaller size sediment. Coral is not a rock, right? Coral is a living creature, and it has some ability to remove sediment on its own. The crabs with small sediment didn't really add very much to it. So in this case, we got to vote for functional redundancy. All right. But sediment removal is not what Trapezia is famous for. It's famous for being a protector of the coral. And no one says this better than David Attenborough, so I've stolen a little piece of his film. Oh, hold up. Let's see if we can get some sound on this, because his voice adds a great deal to this. Nothing here. Let's see. There we go. I'll let him say it. I can't impersonate it.
Some corals are targeted by yet more deadly predators.
Yet more deadly predators.
Predators that can crawl in search of their victims. Crown of thorns starfish, poisonous, invincible eating machines. They also extrude their gut and digest coral wholesale.
The ominous music is fantastic.
But some corals have help. Small crabs living within their branches resist these onslaughts and defend their home. From beneath, they launch an attack on the vulnerable underside of the starfish. Even the crown of thorns will retreat from such a determined attack, and this coral is left unharmed.
So they're pretty cool. So I was curious if the same thing happens from the control, right? So we've got blue here and then yellow for both of these. So there was no difference between the two species of Trapezia, but Trapezia as a genus at this size level is very effective at repelling Drupella. So keeping the snails away from their host corals. When we move up in size though, so size of this one species of crab is quite variable. So depending on the size of its host crab, or host coral, the crab can be the size of your pinky nail or can be the size of your thumbnail. So they're quite variable. And when they get bigger, they no longer care about Drupella attacking their colony. So it seemed very size related. The little ones would block attack, the big ones wouldn't. So there's a size function here as well as species. When I looked at Calcida, so again, about the size of your head, responding to medium-sized Trapezia, size of thumbnail, we see drastic differences. So the coral on its own was toast, with more than 70% of the coral tissue removed from the coral skeleton. The two species of crab were both effective at reducing that and quite different from each other. And then something remarkable happened. Kind of horrible, but remarkable as well. We had an outbreak of crown of thorns. So crown of thorns in the movie, one individual. What happens though is sedimentation sweeps nutrients into the ocean, and all of a sudden, all the larvae of crown of thorns are fed. And there's about a 3-year cycle after, say, a logging event on an island in the Pacific, and then this wave of crown of thorns can appear. And we had this happen on Moorea while I was there. So remember, they're up to 5 overnight. Overnight. So you remove the crab one day, the next day your coral is toast. It is gone. With the percentage of corals attacked, out of the 45, when I had crabs, only 8 of the 45 were even attacked at all. Any damage. 29 of the 45 with the crabs removed were attacked within 2 days, and within 2 weeks that percentage had gone up dramatically. So here, I'm saying this is a bit looser, but I'm saying that this is a demonstrating functional diversity in that I only removed the largest species of crab. 5 species of crab in some of these corals. When I look at the amount of tissue lost on those same corals, the results are even more striking. So you have, when you've got your crabs present, the amount of tissue you lose if you're attacked is somewhere around 2 to 3%. Tiny. Whereas if you do not have corals or your crabs present and you're attacked, you can have generally around 64% of your tissue removed, but up to complete mortality of your coral. Done. Wow. Very striking. What's more is that when we looked at corals that were surviving because they had these crabs present, the majority of the reef around them, these photos are not altered, this is what it looked like. So you had this gray dead reef, and then you would have your living Poeciloptera with its big, strong crab defenders, right? And what we found were other species of corals sheltering in the lee of this mutualism. So the crab is defending its coral, but other species of corals are benefiting from this relationship. Other species are sneaking in under the radar. So crown of thorns ate this half of this Porites colony, but avoided this because it was within the reach of the crab. And we found up to 13 species of corals sheltered by this mutualism that had otherwise been completely wiped off the reef. They were gone, except where this mutualism was in action. So, both size and species seem to matter when it comes to trapezia and coral defense. So our biggest ones are the only ones who seem to really block crown of thorns. Medium ones certainly block Colcida, but they don't seem to block Drupella. Small crabs can't defend against Colcida, but they can defend against the smaller snails. So you end up with this very complex relationship based on the diversity of crabs present in the system. Okay, frame shift, Caribbean, closer to home, where algal overgrowth is a major threat. We have a lot of parrotfish removal and crab removal from our waters. Here, Belize, Florida, all these places. And so algal overgrowth has become a threat. I was interested in looking at this, again, with crab species, or a group of species, and I needed something that was similar in size and form and shape. So I chose this genus Mithraculus. And Mithraculus is something that you have here. I'll bet a bunch of you have seen this little guy. So Mithraculus is a diverse genus. We have 7 to 12 species, depending on which expert you talk to. But this one, Mithraculus sculptus, frequently appears in the aquarium trade. They all eat algae. And if you look at the chelae, or the claws, and the tips, they're actually shaped like little spoons. So this is an adaptation to scraping algae off of rocks. These are purely algae-feeding crabs. They're not known to have symbiosis with their hosts, but there is a range of co-occurring species in the same size range. So I wanted to ask the same question again. Diversity or redundancy? And this was done on a small island off of Belize called Caribou Key. Very small island. So this is a picture from my kite that I put up. And we have a little lab and then 2 dorm buildings. That's actually the outhouse. And this is a really lovely seagrass bed and Porites stand. So that area, that Porites stand, is this finger Porites, Porites divercariata, and it grows really dense in some areas. And it was really interesting because you'd have this very, very dense patch and then nothing. It would just open back up to seagrass or nothing at all, sand. And I was curious, all right, what is making this coral grow so densely in this area? And so you swim around, right? And within that thicket, the coral stems, if you will, the coral branches are covered with coralline algae. Outside of the thicket, when you find the same species of coral, it's got fleshy macroalgae growing on the stems. That's an interesting difference. So you look a little closer, and what you see is this guy, Mithraculus sculptus, all over the thicket. So everywhere throughout this thicket, you go, you find the crustose coralline algae on the stems, and you find little green crabs. You go outside of the thicket, no little green crabs, no encrusting algae. So I had to measure it. So I went out and did these transect lines. So I chose the thickest part of the thicket, or the densest part of the thicket, and then I ran transects going away from the center. And I was looking at stem density and crab abundance, and if it was a coralline algae or macroalgae growing on the corals. All right, I know, graph, little scary. It's all right, we'll get through it. What you need to get from this is that as you go away from the center of the patch, you have reductions in first, the red squares is stem density. So stem density goes way down. The density of those corals goes way down as you go away from the center of the patch. Blue are the crabs. So crab numbers drop as well. And then thirdly, this is the crustose coralline dominance. So in the center of the patch, you have a really crustose dominated stems, right? And then as you go towards the edge, it all becomes fleshy. So all these things are correlated. They're all going along the same trajectory. So one question I had from Trapesia is, do both the coral and the crab benefit? At this point, I was beyond this, right? Because nobody else in this genus even shows up in this patch. So Mithraculus sculptus is in there, nobody else. So even though that they're all, you go flip rubble immediately outside the thicket, you can find 3 other members of the genus, you can't find this guy, or you don't find the same numbers of those other species in the corals. So I was curious, are the crabs effective in reducing the overgrowth of the coral? Like if you actually have an algae problem, will these little crabs stop it? And does stem density impact the quality of the shelter from the crabs? So does this relationship work both ways? Is the crab getting something? Is the coral getting something? So, algal reduction. I conned my friend Michelle Weber into helping me out with this. So basically we got 30 colonies of the Porites. We moved them into this experimental area. And then we put bundles, 25-gram bundles of algae, this fast-growing algae called Dictyota, into each one of those corals with a pair of crabs. So you can imagine this is a lot of fun to arrange. You had to weigh wet algae, you had to catch all these crabs, you had to put everything. She was a really good sport. Thank you, Michelle. And then we left them for 4 days. Okay, another graph. Without crabs, with crabs. Without crabs, and you were looking at reductions. Without crabs, we saw no real reduction in the amount of algae present. In fact, we saw a gain in algae. So the algae was actually growing onto the corals. So without crabs present, the algae will grow onto the corals. With crabs present, we saw a range from just about nothing down to more than 10 grams. So 4 days, itty bitty little crabs eating 10 grams of algae. Significant reduction in algal overgrowth for our reef. And so it all kind of tied in with this idea, all right, the crabs are actively reducing the macroalgae in this thicket. But does it work the other way? Do we get chicken and egg? So I was curious about quality of shelter. What are the crabs benefiting? Well, one thing, as you would know from swimming around out here, is the world is filled with little wrasses. And wrasses will eat anything that you give them the opportunity to eat, right? So if a crab is exposed, wrasses will eat it. And they're annoying as you're a collector because you'll flip over a piece of rubble, you'll say, oh, that's a neat whatever. You'll go to get your jar, and by the time you've turned back, whatever it is you saw is completely gone because that wrasse just snagged it and has eaten it. So they're a very effective small predator in the system. And all I would do was create artificial colonies with varying stem densities. So I'd put 5 stems together, or 15 or 25 branches of coral together to create an artificial colony. I'd put in my crab, and then I'd just time it with wrasses everywhere around. And again, the results were very striking, different colors. So when I had 5 stems, and this is, let me back up. This is an odd measurement. So this is log seconds because the real data wouldn't fit on this graph. The real data, the average for 5 stems was 9.5 seconds. The crabs were like, this place sucks, I'm out of here. For 15 branches, it was 332.6 seconds. So that's about 5.5 minutes. So a lot longer, significantly longer. But when you go up to 25 branches, you're up to 40,320 seconds, or at least 11.2 hours. And the numbers for this were actually slightly greater because I got bored after 12 hours of watching corals. And so I would just, all right, you win, you're staying. I left it at that point. Come back 24 hours later, many of the crabs are still there. Okay, one last group here, host matching. Another frame shift, still Caribbean. This is, I know, having been diving with some of you over the last couple of days, I know that these anemone shrimps are a great draw for a lot of you. I was working on this one called Periclemenes yucatanicus. So it's a spotted anemone shrimp. And this shrimp is interesting in that it is frequently found on 2 different hosts, both on the corkscrew anemone that we frequently see on the dives here, but also on this sun anemone. And so I was curious, all right, are some of the hosts better? We know some of the symbionts are better for these things when we're talking about functional diversity. Are some of the hosts better? And so I went out and I would get gravid Periclemenes. That means that they're holding eggs on their swimming legs. So these female shrimp, I'd go out, I'd get them from their anemones, I would measure both the anemone, the shrimp, and then I would irrigate the eggs from the shrimp. So you can imagine trying to keep this very delicate shrimp perfectly happy as you're flushing the eggs with a syringe from her swimming legs. It was a little tricky, but we got all the eggs, and we got some interesting data. Egg number has a directly linear relationship with shrimp size. That makes sense. That's true of most crustaceans, it's true of most fish. You have this, as shrimp size goes up, egg number goes up, right? But when we look at the shrimp in the 2 different anemone species, so in the red, we have shrimp in the sun anemone, and then in the blue, we have shrimp in the corkscrew anemone. And they form very different groupings, despite the anemones having at least roughly the same size range. So you get up into the 20 centimeter range for these things. 2 different responses based on host. So again, it's a hint of functional diversity. So, there are two things that go into this biodiversity, ecology interaction. One is taxonomic detail. You need to know your organism. And the second is ecological studies, like I've been presenting to you tonight. And together, maybe we get a better large-scale understanding of the ecological importance of biodiversity. It's important. It's important in even human finance. This is a paper that came out not long ago in one of the two most prestigious science journals that we have. This is in Nature. And this is saying that the more nodes that you have in a network, this furry network up here, is all different nodes of banks. And what they found was banking systems were more stable with more nodes. So if there was more interconnectivity, more interaction, banking systems were actually more stable. So what we're learning from ecology is actually being directly paralleled in finance. But what about that whole taxonomic detail issue? And it starts with a very simple question of what species do we have? Like I mentioned earlier, currently we have 192,000 described species of macrofauna. But estimates are at least 1 million, which puts us at 20%. If this changes to 20 million, you can imagine how low on that totem pole we are at this point. So I've been a part of several different projects looking at global biodiversity. This was the Moorea Biocode project. And we have various different ways of going out. Most divers would consider what we do horribly destructive, at least visually. But what we are able to find are all the little things that live in the reef. So you go out and you pick up a piece of rubble and you brush it into a bag. Or you will hand collect. This is Gustav, who will be coming in later on in the Sea&Learn series. This is me digging a hole through the rubble into the reef. I frequently will freak out my dive partners because I'll be torso deep into a hole in the reef. And this is actually an underwater vacuum cleaner. So there's a reef matrix. The living reef is just a thin skin, and beneath it is where things get really interesting. But it's impossible to get into. So I dig a hole, and then we come along with a vacuum cleaner and can actually remove some of the small animals from within that reef matrix to figure out what they are. In Moorea, we were genetically sequencing all of them. And so the goal was to sequence everything on the island. Ambitious goal. A more approachable way is perhaps looking at 1 cubic foot. So David Lichtwager from National Geographic has been going around to different environments and putting in 1 cubic foot, and he came to Moorea to work with us and put in this one at Tamai Beach, right here on the corner. And so this is just 350 individuals from that 1 cubic foot. But we had over 600 individuals from that same area of about 300 species. We're still figuring out exactly the species count. 1 cubic foot of reef. Of those individuals from the 1 cubic foot, we had 26 gastropods, 24 fish, 13 echinoderms, 26 worms, 7 heterogastropods, and 60 different species of crustacean. 1 cubic foot of reef. So, my question to you is, how do you get involved? The first step in understanding a fauna, as diverse and as remarkable as Saba's is to know what species are here. And so, as a thank you present and as an opportunity, I prepared something for you. Let's see if I can get it to work. It's a little set up in a program called iNaturalist.org. And this is available to all of you on your computer for free. It's available on your smartphone as well as an app. And what it allows you to do is upload your photos and information of your sightings. So if you were to go out diving tomorrow and you take a picture, you can upload it to the Biodiversity of Saba now online later on that afternoon, later on that year, or if you do it with your phone or you You can have it geo-referenced, so it'll tell you where on Saba it is. And let's see if we can get it.
See Saba.
Let's see. She's skating. Keep going to your right or left.
You'll need to zoom.
All right, go to the other side. Keep going. There you go. Oh, I think you're going to have to drag the window onto the screen for us. Thank you. All right, so this is online now. And so if you go up to that projects, once you sign in to your iNaturalist, and then pull down, you should go to Biodiversity of Saba. We click on this, and hopefully it'll open up for us. And this is up and running right now. So you are absolutely welcome to start adding to this today, tomorrow, whenever you feel like it. But with the experts that you have coming in through Sea&Learn, you should be able to do a census of Saba's marine life and terrestrial life. I mean, the orchid crew could do this tonight. I mean, they love working really hard. So you can add in all sorts of things. And people have already started adding stuff in. So I created this about 2 hours ago. And people over the world are already adding stuff in. So Sea Kangaroo, who's an observer who participates in iNaturalist, has already added several photos of this snail, although it's unidentified. There's a green iguana, the Anolis species here. It's really easy to do. You upload your photo or your information, and this is now available to you. If we can go back to the PowerPoint or the keynote, I know that's a pain. All right, and the next slide, I can take it from here. So, conclusions. Both functional diversity and redundancy are observed. Scales of diversity, including species, size, behavioral diversity, can have legitimate effects on reef survival. And we need to know more about both species identity and the ecological relationships if we're to have an effective understanding of marine biodiversity. So with that, I'd like to say thank you very much, Saba. Thank you, Sea&Learn. Thank you, Yelp. Thank you, all these other people who've helped me out along the way. I really appreciate it. And I'd love to take any questions.
Okay, so from this we can draw that crabs are a friend, we think. They're helping to control algae and sediment issues.
Well, some species, yes.
So, has your research shown any indicators of what are specific threats to crabs, or just their
That's a really good question.
like everything else?
All right, so Trapesia, at least, the defender crab, we know it drops out with high temperature. So if you raise temperature on reefs just a few degrees, Trapesia no longer survive. It drops out with decreased salinity. So a lot of these things that we associate with really inshore, mucky reefs will wipe out Trapesia. Mithraculus, no one's done that work yet. A lot of these are open questions for many, many taxa. Yes?
Are crustaceans spineless invertebrates?
They are. They wear their they have an exoskeleton, so they have their skeleton on the outside. So it's like having a suit of armor instead of a skeleton. If you can imagine, you know how your muscles attach to your bones? Their muscles attach to their armor. It's a completely different way of doing things, but it's worked out really well. They rule the world. Yes?
Have you done any research on pea crabs?
I have not. It's a fascinating group, and luckily there are people working on pea crabs because they're showing to have an impact on mariculture of different bivalves. So raising pearl oysters, for example, or in the Pacific Northwest, clams to eat. Pea crabs are having some interesting effects, so there is some research going on. Fascinating group. Yes?
You only did 4 dives on Saba, so it's not an easy
insufficient data.
He wants to come back.
I would love to come back. Thank you. Any other questions? Yes.
The coral bleaching, we're all concerned about. That affects the crab populations too?
Coral bleaching is interesting. So the crabs are actively being fed by the coral. So if the coral is unable to produce its own food, the first thing to get cut off the list is its army, right? So the crabs at that point either bail or they die. So if the coral can regain its endosymbionts relatively quickly, then the crabs will linger for a week or more before they leave. But if the coral doesn't recover rapidly, then the crabs are done. And you have to wait for the next wave of recruitment for the crabs to have any protection.
Lifespan of some of these crabs?
Another excellent open question. I know that I have followed individuals for 2 years of the biggest species to decrease salinity in those environments, so I don't know what the nutrients themselves are doing. Open-ended. More research to be done. Yelp kids, this is your cue. Thank you again. I really appreciated being here.
Let's give a big thanks to Dr. Seabird McKeon. We would also like to thank the Johnson family and May and the staff here at the Cottage Club for hosting us tonight. Thank you to Paul and the staff at Shearwater for hosting Seabird on his stay. What's coming up? Well, before I get to that, the two little boys that are here, if you see one of the two beautiful Sea&Learn girls, they'll give you a game of Uno. Compliments of Dr. McKeon. Is this your real name? Yes.
Insufficient data.
Okay, what's coming up on Sea&Learn? Thursday night, we move up to Momo Cottages, where Dr. Tammy Goulet will be talking about the symbiosis at sea. And Dr. Goulet is here somewhere. Wave your hand. There she is back there. We were out and did a dive with Dr. Goulet today with the Yelp kids, and it was a lot of fun. And if you really want to work with a scientist underwater, on Friday afternoon, we do have a field trip going out with Dr. Goulet. It's really pretty interesting stuff. On Saturday night, we move to Lambie's Place, hosted by Eden, where Dr. Martin DeGraff will be our speaker, and he's going to be speaking on the fisheries on Saba and Statia, and in particular the Saba banks. I'm not going to go much beyond that because we're only at the halfway. But do circle October 30th on your calendar, because closing night of Sea&Learn, we're going to rock your world. If you're not here, maybe you should extend your vacation and stay here for a while. Okay? Thank you very much. For those people that left beer bottles on the wall there, please be friendly and put them in the garbage for us. Thank you very much. See you all Thursday night up at El Momo Cottages.